So this is a PowerPoint lecture for Chapter 15, finishing up our Unit 7 on Mendelian genetics. And so what we're going to focus on here is going to include some of the errors in meiosis and the role of the chromosome in the functioning correctly of genetics. So first of all, what we are looking at here is a chromosome. And so where Mendel's heredity factors were actually located, he didn't know what they were. He didn't know anything about genes or chromosomes. But what we know now is that the factors that he was studying, the different characters of his pea plants, were actually being expressed and controlled by genes, which are located on chromosomes. And now we know we can actually find a particular gene as seen right here, and where it's located, its locus, by tagging it with a fluorescent dye that highlights that gene. So what you're seeing right there is an actually isolated gene that's been tagged. So looking here at the chromosomal basis of heredity, if we look at what is going on specifically in a cell, based on what, oops, that didn't really help you guys. There we go. Based on what Mendel was studying, if we zoom in on the nucleus and actually the cell as the nucleus dissolves, we can see here one of the things that he studied, such as the shape of the seeds. And so we can see the yellow round seed and the green wrinkled seed. And now we know that those are two characters that have two different alleles. There's the Y, dominant Y, and the recessive Y for the color. And then we have the dominant R and the recessive R for the shape of the seeds. And then we can follow through meiosis what occurs. So if we have the formation of gametes, we have the maternal side, we'll say that's the red side there, forming a haploid gamete with one set of chromosomes. And so that one set, it's one of the homologous pairs from the mom's set of genes. And then the blue side we'll say is the dad's or the paternal side. And again, we go from a diploid paternal plant producing a gamete that is haploid there with two chromosomes instead of the four it started with. And then we cross that parental generation, and when we have the joining or fusion of those two gametes, we go back to diploid there. So you can see after this line right here, we have our F1 generation. And so now we see the combining of these two sets of chromosomes. So now we're back to having a homologous set of chromosomes. And then here we have, again, meiosis occurring. And during meiosis, what this one is showing, we have the occurrence of the law of segregation, where the two alleles from each set of genes separate. So basically what we're having here is the breaking up of the homologous set of chromosomes right here. And we have one set going one direction, one set going the other. But at the same time, during metaphase 1 and anaphase 2, we also have the law of independent assortment occurring, where we have them lining up in different orientations. And that's an independent assortment of how they line up on the metaphase plate. So you can see this set of lining up chromosomes is different from this set over here. And that results in our daughter cells having a different makeup, again, of possible chromosomes. And then we go through meiosis 2 and end up again with our different gametes. So this is important to think about. So when we're talking about what is actually going on when we are crossing just the letters of a set of chromosomes, what actually are we crossing? And what actually is occurring through meiosis to those different alleles. So remember always that those letters are representing alleles, and they're part of the homologous set of chromosomes. So this slide is actually talking about how Morgan did a lot of work with fruit flies, and you got a chance to do that with your lab. But one of the things that Morgan determined, he actually determined quite a bit. He learned a lot. And one of the things was that the white-eyed mutant allele had to be located on an X chromosome. And his findings supported the chromosome theory of inheritance. So what he did in crossing this set of chromosomes here, he had his female with this set of chromosomes, and then this set of chromosomes here for the male. And he found that just with the results that the F1 generation all had red eyes, but then the F2 generation showed a 3-to-1 red to white eye ratio, which we would have expected. We expect a 3-to-1 ratio, but what was different is that only the males ended up having the white eyes. So that's where we start to see when we have little differences like that occurring, or actually that's a big difference, then that tells us that there's something else going on, such as this being an X or sex-linked trait. Morgan's discovery of a trait correlated with the sex of the flies, and the fact that there was a correlation with the sex of the flies or the gender was a key to the development of the chromosome theory of inheritance. So in humans and some animals, there is a chromosomal basis of gender determination or sex determination. So we have two varieties of sex chromosomes. We have a larger X, which you can see in that picture. There is an actual photograph of one, and then the smaller Y next to it chromosome. And you can tell by looking at that picture that those chromosomes are actually, they've already gone through interphase and they've been replicated to be sister chromatids at this point. So a person with two X chromosomes, as you well know, is going to be a female, and a male is going to have one X and one Y. And only the ends of the Y chromosome have regions that are homologous with the corresponding regions of the X chromosome. This slide shows you how we have our XY system in humans and some animals, but there are other systems as well. So you can see here that our XY, we have 44 chromosomes that are autosomal, and then we have our sex-determining chromosomes, either the XY or the XX. And then when we have these cells go through meiosis, we end up with gametes that still have the autosomal chromosomes. This time we've gone to half of the 44, so we have 22. And then we have one of the sex chromosomes. So in this case, the sperm are going to either carry an X or a Y chromosome, whereas all of the eggs are going to carry an X chromosome. And then when there's fertilization, we go back to having our diploid zygote. And the zygote again goes back to having 44 of the autosomal chromosomes. And if we combine this X with this X, this sperm with this egg, we end up with a female. Or if we combine the egg with the Y sperm, we end up with a male offspring. So again, short segments at the end of the Y chromosome is homologous with the X chromosome. So that allows the two chromosomes to behave as if they are homologs, even though they really aren't, during the meiosis in males. And that allows the male gametes to carry both the X as an option allele or the Y allele as an option. There's a gene on the Y chromosome that's called the SRY, which is the sex determining region on the Y. And that's what is responsible for the development of testes in an embryo. And so that is what actually has the embryo develop into a male. And a gene that is located on either sex chromosome is called a sex-linked gene. If the genes are on the Y chromosome, we call them Y-linked. And if they're on the X chromosome, they're called X-linked. But X chromosomes, as you saw, are much larger, and so they have genes for many more characters that are not related to sex than the Y chromosome does. Most Y-linked genes, so genes that are located on the Y chromosome, are related to sex determination. So this is showing again how we have the production of our different gametes that are just simply sex-linked. So this is how we would follow the transmission of a X-linked recessive trait, for example. So if we look here, we have a homozygous dominant for the trait, and this is important to study this graphic, because it shows you how to use this sort of representation of chromosome alleles and their actual difference in alleles. So what I mean by that, I wish, that this was a arrow that I was using to point with instead of a cross. But what you can see here are those two X's have the capital N's, and so they are showing a dominant trait. But here we have the X that's associated with the male here is showing the recessive trait. So as we have this cross occur, we can make our Punnett square where our male alleles are here. We have the X with the recessive trait, and then we have the Y that doesn't have any trait at all. So this is telling us that this is only found on the X chromosome. And then we have the eggs where it's both of the gametes, the possible gametes, have the dominant allele. And so we end up with this cross right here. And in this cross, all of the females are going to be heterozygous for the X-linked trait, whatever that was, this recessive condition. Whereas now all of the males in this F1 are going to not have that trait. They have the dominant, or probably what we would call the healthy version of this allele. So now here we take this female and we cross it with this type of male. So we have our heterozygous female now creating our possible offspring with a dominant traited male here. And again, you can follow through and see how our different gametes are formed and what we end up with offspring. And you can see here now we have a male that is showing that original condition with the recessive trait, and one female that is a carrier, and then two that are not showing the trait at all and have no alleles for that trait. And if we cross the heterozygous female with a male that actually has that trait, then we're going to end up with half of the offspring having that possibility. So both a male and a female offspring will actually display that condition now. The other female will be a carrier, and the male will be the only one without the trait at all. One other fact that we need to talk about is that in females, there are these two X chromosomes. So